Hi everyone, welcome to this EMQN guest webinar for 2023. Thank you so much for joining us. I'd just like to start the session with a few bits of information. So please be aware that we are recording this webinar and we'll be disseminating the link to all registrants shortly after the event. And for this reason, we kindly ask that you refrain from using any patient or EQA laboratory identifiable information during any discussions today. I'm also really pleased to let you know that this is a CPD certified event and we will be circulating certificates to all attendees of the live event shortly after close of session today. And I'm obligated to say that the views expressed in the webinar are those of the author only and do not necessarily reflect the views of EMQN. So we are hoping to hold a short Q&A session shortly after Heidi's webinar. And so if you would like to submit a question, please use the Q&A button, which is available in the banner at the bottom of your screen. And with that, I'd like to hand over to Dr. Simon Patton, who will introduce our speaker today.
Thank you very much indeed, Rachel, and welcome to all of you, and thanks for dialing in. We've got a hopefully, well, no, not hopefully, but a very insightful presentation today by our guest speaker. So without further ado, I will introduce Professor Heidi Rehm, who is the Chief Genomics Officer at Massachusetts General Hospital and co-director of the program in Medical and Population Genetics at the Broad Institute. Her work is focused on discovering the causes of rare disease and building resources to support the integration of genomics into medical practice. So Heidi, the floor is open to you. Thank you once again for presenting, and we'll look forward to your presentation and the Q&A answers session afterwards. Over to you.
Terrific. Well, it's a great pleasure to be here and to be able to share with you some of the work that we are doing. I'm going to share my screen. Let's see the screen. Okay, I think we're all set. Hopefully you can see my slides. And I'm just as a comment, I'm happy to share these slides with anyone who would like a copy after the talk. Thank you. So I'm going to talk a bit about how we are working to advance genomic medicine through collaboration on a global scale. There we go. So I did come up with 3 questions just to get a sense of the community that I'm speaking to today in terms of EMQN. And I asked 3 questions. One, do you or your laboratory submit classified variants to ClinVar? And so this is the responses we got. So over half either are already or plan to, so that's great news. But there's still a good chunk there that does not submit. So I encourage you, hopefully maybe after you've heard my talk today, you will consider it. I also asked if you're participating in ClinGen expert panels. Most of you are not, but a little over a quarter of you either are or are planning to, so that's great. And I'll talk to you a little bit about what those panels entail and an opportunity to get involved and why that might be useful. So, and then the third question I asked was, do you or your laboratory use Matchmaker Exchange? And it looks like, close to a quarter of you do. And another set, it's not applicable because you're not actually performing exome or genome sequencing, but a lot of you aren't. And I will, at the end of my talk a little bit about the Matchmaker Exchange, and maybe you'll find that it could be useful for some of your work. So with that, I'm going to dive into talking a bit about our Clinical Genome Resource project. So this really has a goal of creating authoritative central resources to define the clinical relevance of genes and variants for use in both medicine and research. We started it now 10 years ago, and it's primarily funded in the US by our NIH, and it has 3 core U41, which are these genomic resource grants. But now a number of the NIH institutes are now funding disease-specific expert panel grants to help expand the work that we are doing. And in a nutshell, we really are focused on aggregating genomic and health data from different sources, patients, clinicians, labs, researchers, and then curating that information to define gene-disease relationships or variant pathogenicity or dosage sensitivity, in particular, whether genes or chromosomal regions, when duplicated or deleted, lead to a phenotype, and then clinical actionability. So if I had sequenced someone's genome and had a particular variant, should I return it? Is there some action that could be taken that would make that clinically useful? And so those are the types of curation we do, and then we disseminate that curated knowledge to the community for use in many different ways. We are also really at this stage, we have 2,200 investigators across 56 countries that are participating in our expert panels and working groups. So it's really a large community effort. And in terms of the databases and where this information is going, I define two databases, ClinVar, which is really a database for sharing variant level knowledge, which is supported by the NCBI at NIH. And then we also have a newer database I'll talk a little bit about, which is the GenCC, part of the Gene Curation Coalition. And that is a database for sharing gene level knowledge that's supported by ClinGen. And so any laboratory can submit variant pathogenicity claims into ClinVar or gene disease relationships into GenCC. On the other hand, ClinGen is really working at building an infrastructure to support expert curation. We use tools like our variant curation interface, our gene curation interface, or in the somatic cancer world, the Civic database. Now, I do want to point out, why do we curate both genes and variants? So I think it's useful to think about how we implicate variants in disease, which is what a lot of us do. And you may, be familiar with one of the most common pathogenic variants, Delta 508 in the CFTR gene that causes cystic fibrosis. So in this case, there is both variant level evidence. This variant we all know is pathogenic, but it's also, there's gene level evidence. We all know that the CFTR gene is the gene involved in cystic fibrosis. However, for any gene that we know is involved in disease, we routinely still find variants of uncertain significance. So the gene's involved, but we don't know what the variant means. We also, of course, there's still many genes that we don't know what their relationship with disease is. And so, as we sometimes say, you find vooses and gooses, or a VUS and a GUS. But one thing that's worth pointing out is you cannot have a variant that is pathogenic if the gene has never been implicated in disease. So it's a really important first step to define those gene-disease relationships before we can really say a variant is pathogenic for a given disorder. So one of our key first steps is doing this gene-disease validity curation. And we developed a framework through ClinGen that really assesses all of the different types of evidence for implicating a gene in disease. And that includes gene-level evidence. Was it observed in cases with disease? Did it segregate through a pedigree? Was a case control study done, as well as experimental evidence? Where's the gene expressed? Does a model organism demonstrate the disease? Can you rescue the phenotype with replacement of the wild type gene? All these types of studies. We evaluate all of that evidence through a semi-quantitative framework where points are scored for different types of evidence, and experts review all of that data in our expert panels. And then we end up classifying the gene into one of these tiers: definitive, strong, moderate, limited, animal model only, no known disease relationship, or we're disputing or refuting a claim that's been published. Now, once we classify genes into these categories, we then, many of the clinical labs use this information to define what genes are used in clinical testing. And so the American College of Medical Genetics and Genomics released a guidance as to how to use these different tiers in terms of putting genes into tests, with the basic ideas that genes that score moderate and above could go into a disease-specific panel test, where the clinician is ordering a set of genes known to be involved in that phenotype, and therefore you should really only include genes that have at least moderate evidence for a role in disease. On the other hand, if you're doing exome and genome sequencing, including the limited and below is fine. We're looking at every gene in that case, and so these limited genes and below will be included and looked at there. Although what we report back to patients may be based on the actual evidence level. So ClinGen has been doing this type of curation now for many years through many different expert panels that have developed over time. We have data on our website that defines all the different categories of curation and the results. And so to date, we have curated over 2,000 different gene-disease relationships. And this figure breaks down where we are with the percent. They're in what I consider the insufficient evidence categories, so limited and below. And what we find in terms of what's published in the literature is about 25% of gene disease relationships that are published don't have sufficient evidence to support that claim. Not on this slide is the fact that we've since been revisiting these genes over time, and I can tell you that most of those gene claims that have been published but score limited below never actually evolve. A small subset of them do change, particularly the more recent ones, but a lot of these just simply never had sufficient evidence and will never have sufficient evidence. I think the most startling example was when our expert panel on arrhythmia disorders curated those genes involved in Brugada syndrome, or short QT, and they found that nearly every gene, 19 out of 20 were disputed and didn't have sufficient evidence for a role in disease. And one thing that's a trend as we look at the different types of evidence that support gene disease relationships, on this figure from our cardiomyopathy group, you'll see that a lot of the genes that score limited and below here in the middle section really primarily only have experimental evidence and very little case observations. And that's a trend that we see where, one or two cases will have a missense variant. Somebody thinks it's, implicated in disease, and then they'll make some sort of functional system to try to prove that gene's involved. But those functional systems don't always recapitulate true human disease and may be a red herring in terms of that gene's role in disease. So beware, if there's not a lot of cases published, sometimes that can warn of limited evidence. Now, if you're interested in looking up the state of a curated gene, ClinGen maintains all of our knowledge base on our website. This is a screenshot. You can go to the website, you can type in your favorite gene and see if it's been curated for any of the types of curation: gene disease validity, dosage sensitivity, actionability, variant pathogenicity, or pharmacogenomics relationships. You can either search for all the different types of curation, or on the right here is just going to our gene disease validity lookup and looking for genes there. Now I'm showing, if you click on a gene and bring that up, you can see all the different types of curation. And that will also highlight that sometimes a gene has been curated for its involvement in multiple different disorders. So in this case, the ACTC1 gene has been curated for its involvement in both hypertrophic, dilated, and arrhythmogenic cardiomyopathies. And you'll also see that the conclusion of the expert panel differs across these three different phenotypes. So definitive for HCM, moderate for DCM, and finding no evidence for its role in ARVC. So it's useful to keep that in mind, that multiple claims can be made on a gene, some of them valid, others not so much. Now, how are clinical labs using this information? So here's an example. Invitae, a large genetic testing lab in the US, follows our curations, and they define their panels with a primary panel that only has genes that have been curated with moderate evidence and above, but they do allow clinicians to manually add on other genes if they wish to. And so most don't. They usually order just the primary panel, but this is a way to deal with some of the sort of lower evidence genes, and the fact that labs sometimes compete on how big their panels are, so it's offering options to all physicians. It's worth pointing out that if you've scored a gene or one of our expert panels, moderate evidence, then really there's a cap on the level of variant evidence that you should be scoring. So for moderate genes, likely pathogenic is the cap, whereas for limited and below, you wouldn't interpret a variant as anything but variant of uncertain significance. Now, I mentioned this database, the GenCC, for the Gene Curation Coalition. So this database, we started this effort early on when we knew there were a number of public resources like OMIM or the Genomics England panel app or TGMI, different databases that define gene-disease relationships. And we were trying to get all those databases to harmonize the terminologies and the approaches they were using so that these databases could be interoperable. And so we set out to develop consistent terminology, and then also with the goal of publicly sharing, because we then discovered there were a lot of clinical labs that were also doing curation of gene disease relationships to inform the panels that they were putting out for genetic testing. And so on the right side are a lot of clinical labs that joined this effort whose data wasn't in the public domain, but they're willing to share those gene disease curations that they had done. And then the goal that we had as the Gen CC was to collaborate across all these groups and resolve any differences. If one group said, oh yes, this gene is implicating disease, it's in our panels, and another group said, no, that's limited evidence, we needed to resolve those differences so that we could all work towards a harmonized curation ecosystem. So we set off, and first we collected all the different terminologies and evidence levels that were being used across the public resources. And what you can see here is that we were all over the map, different terms. Two groups are using color schemes, but they were oriented in the opposite directions, with red meaning definitive in one database and red meaning disputed in another database. So you can see how this would create confusion for the community trying to use all these different tools and resources. So we went through 3 rounds of Delphi surveys with the broader community, all of these groups, and then turning externally to the user community to really come up with a common set of terms. Ultimately, we ended up using the ClinGen framework with the addition of the animal model only term to extend that, which we've now adopted within ClinGen as well. And so this became the framework that now all of these databases can map their terms to, or some of them have moved directly to this new system. We also then brought in all of the data from these different groups. We now have 12 different submitters representing over 16,000 different gene disease relationship curations on over 4,600 genes. So this is a screenshot of the interface that we've built for anyone to use. I like to call it the ClinVar for genes. In the same way labs submit their claims on variants to ClinVar, you can submit your claims on genes to GenCC. And here I've just clicked on the GJB3 gene on the main search interface, and then you'll quickly see different submissions. And for every submission, they're defining the evidence level and the disease that claim is on. And so you might see, in this case, 2 groups saying strong for a skin disorder, but 2 other groups saying disputed for the hearing loss claim. So in this case, they're actually harmonized and equivalent, but they're doing different claims on different disorders. We have monthly meetings of this group now that we're all sharing this data, and we can define those discrepancies. To date, we've resolved 21 different discrepancies in gene disease relationships, and we really try to just resolve them at the level of moderate and above, or limited and below, because that really informs whether labs include or not include these genes in disease-specific panels. Although we have a couple of genes that we all agree were just so borderline, we couldn't resolve that. Okay, so that's sort of a lot of the work we're doing across the community in gene disease relationship definition. And we hope within ClinGen to curate every gene disease relationship that's been reported. On the other hand, if we turn and look at variant classification, it's a much more daunting task. We have billions of variants across our genomes. There is no way that any expert panel is going to curate all the variants in a given gene or across all the genes in our genome. So we take what I call a sort of 4-pronged approach to tackling the variant pathogenicity challenge. One is that we work on developing common standards and disseminate those standards across the community, both in terms of terminology and rules for variant classification. And probably many of you are familiar with the first version of the, well, actually it wasn't the first, it was the 3rd version from ACMG, but it really defined a framework to apply terminology and define that terminology and define evidence types and how to arrive at those curations. So this was the framework that we published back in 2015. You may be familiar with this little rainbow chart that I had made as part of that to define all these different evidence types across different categories and the terms that would be used. As soon as that was launched, ClinGen formed its own sequence variant interpretation working group that's led by Les Abisaker and Stephen Harrison. And the goals of that group were to continue to refine and evolve those guidelines as they were tested and deployed by the community. And I'll share some of the work we've done. We also, all of our expert panels are then specifying this general framework for their disease or genes that they curate on. And so we have a group that reviews all of those specifications to make sure they're within the bounds of the framework. And then we've been gradually working to move this framework towards a more quantitative approach. And so you'll see both white papers and published papers on the ClinGen website where we've given additional guidance for how to evaluate evidence for pathogenicity. This is just an example of more specific guidance around the de novo observation type rules that's on our ClinGen website. And actually, this is a screenshot of the ClinGen website. If you're looking for these additional types of guidance, there's a list of the different codes that we're providing additional guidance and the links to publications or white papers that have that information. Also, I've pasted onto the right side of this slide. We now have launched a repository of all of the specifications for all the different genes. Our expert panels develop these gene or disease-specific specifications, and you can go in there and search for any gene or of interest and get those gene-specific rules. As some of you may be aware, we have been working diligently on a new version of the ACMG guidelines for variant classification. And we actually presented a draft version of this guidance at the ACMG meeting last month. And there's a link on this slide at the bottom that gives you a sense of where we're going, but just at a high level, some of the key distinctions is we're now moving to a points-based system. It's actually based on the Bayesian framework that was published and developed by Sean Taptijan and our ClinGen SVI working group. And so we'll be using that as a foundation for this next version. Also, we'll be using, giving guidance on the sub-tiering of VUSs. There's a very large range of confidence for pathogenicity for VUSs. And so we will work to subdivide that into 3 different tiers, low, mid, and high in terms of the evidence level. And then we also, actually, I'm co-leading a working group through ACMG that will then provide additional guidance for how to use those sub-tiers in terms of reporting variants back in clinical reports and how we use them in the community. So that's one approach. If we can disseminate the standards to the community, then all the variants that are curated by our global community will be more likely rigorous and accurate in their interpretation. That said, we know that there's subjectivity to applying these rules, and sometimes we arrive at different conclusions, or sometimes we have different evidence within our own laboratories. So it's really important that we all share our variant classifications publicly. It creates transparency and also crowdsources the work. And just as a specific example of the benefits of public data sharing, in July 2017, a couple approached me because I had submitted a variant through ClinVar, and they had received a genetic test report with this variant of uncertain significance. They were, of course, concerned about their pregnancy and asked if we had any other information. So I reached out to the other lab at GeneDx that had also observed this mutation. We gathered the clinical data across the 2 laboratories, and we were able to reinterpret that variant as likely benign with all of that data together, and then updated the classification, which is now updated in ClinVar. So it really shows the importance of identifying, even if it's a variant of uncertain significance, that your lab has observed that. And we often collaborate across labs to share evidence to reclassify variants. So to date, there's over 3.3 million submissions that are within ClinVar on over 2.2 million variants. And those submissions come from all around the world. I will note that about 96% of this data in ClinVar is actually from clinical labs, with a smaller fraction from researchers. And that's in part due to a lot of effort that we developed early on to engage with clinical labs, encourage and support them in data sharing. We no longer financially support them through our grant, but all of these labs voluntarily share that data through ClinVar. And that, because of the high volume of classifications happening in the clinical setting, it represents the largest portion of what's in ClinVar today. Now, I mentioned early on that there's sometimes subjectivity or to classifying variants, or different internal evidence that each lab may have. And therefore, there are differences in the variants that are classified. And so we've expended effort within ClinGen to resolve differences in variant classification through our interlaboratory efforts. And if you look at ClinVar, you'll see that there are thousands of clinically significant differences through those submissions. It represents only about 1% that are these medically significant differences, but nonetheless, resolving these is important. And we've done a number of different efforts to resolve these classifications. In different phases, and these have been published by Stephen Harrison and colleagues in different efforts over time, where we've been able to resolve a lot of these different variants that have been classified. And we even, in this right panel of this slide, we're able to exercise this variant harmonization effort within the All of Us research program that's returning results to patients. And we were required by the FDA to harmonize so that no different results return to different participants in that program. And we were able to resolve 100% of the discrepancies before we initiated return of results. And now we have an ongoing variant adjudication committee that resolves any differences over time. Now, occasionally within our general community of laboratories, we aren't able to resolve the classifications, and that's when our expert panels are really critical in this process. So we have expert panels that span 13 different clinical domains. You can go on the ClinGen website and see all of these different clinical areas that we manage variant curation expert panels. And of the 59 that we have today, 22 of them have full approval to begin classifying variants. And they do go through a rigorous process of defining their work group, conflicts of interest, defining the scope. Then they develop the rule-specific classifications, and then we pilot they pilot the rules on a set of variants, each time interacting with our sequence variant interpretation review committee. And then once that is reviewed and approved, then they can go off and start classifying variants. Once they do, they submit those to ClinVar, and they get the FDA recognized stamp because our whole protocol has been reviewed by the FDA. It allows us to have this stamp. In addition, those variants get the 3-star classification in ClinVar, which means they override any of the individual submitter entries to resolve conflicts that may be occurring across submissions. You can go to our ClinGen website and see this stage, and this little green bar on here indicates the status of each of these expert panels in getting approval, and see what they're up to. And just as an overall status across all of these expert panels, close to 5,000 variants have been curated to date. And you can also go to our evidence repo, where all the detailed evidence types and data has been attached to those variants. There are links from ClinVar to get there. And so this gives you a sense of the different curation that's been done across different genes. Now, I mentioned early on when I asked that question, how many of you are involved in ClinGen expert panels, and there's still a smaller subset of this community I'm speaking to today. So I encourage you to get involved, and you can go to our ClinGen website and sign up. And either, perhaps you're a more junior member of the community, you're interested in being taught how to curate variants and participate. So we give that training and we assign you to an expert panel as a volunteer. There's not a lot of curation that you are required to do to be part of that, maybe one variant every couple of months or something like that. So it's not a huge effort, but it gets you engaged, trained, and part of this curation ecosystem. Also, if you are an expert in a particular disease area, you can contact that particular expert panel and ask to join a particular panel as an expert. Then you'll be involved in reviewing the work of the biocurators that are curating for that group. So both are options to get engaged. Also, we have our variant curation interface is open to the public. So if you don't have your own variant curation interface, you can use ClinGen's for free. My own research group uses it, and that allows us also to more easily submit our data to ClinVar. We also have a gene curation interface that is only used by our expert panels because we only want ClinGen to be curating one gene disease relationship per instance. And this whole program is led by an amazing group of colleagues. Shown here on the slide is our ClinGen leadership, our steering committee. But we have over 100 other staff that work very hard in coordination and biocuration, software development, many other roles, and managing the 37 different working groups that manage our standards and our operations and our software to support this whole ecosystem, which of course contains many volunteers. I mentioned 2,200 expert panel volunteers from all around the world that are participating in these expert panels to help create this knowledge for our community to use in the care of patients. Now, the one last piece of my talk I wanted to touch on these limited evidence genes that aren't yet robustly involved in disease. And so in this case, what do you do with one of these genes that scores limited in particular? Well, you need to find more cases, more evidence. And it's worth pointing out, if you're using the OMEM database, the stats on there suggest that that we've only implicated about 24% of genes in the human genome in disease. Yet from this paper I reference here from my colleagues at University of Washington, there is evidence that there's probably over at least 5,000, probably closer to 10,000 other genes yet to be implicated in disease. So and we daily are implicating new genes in disease. This number changes constantly. So if you're a clinical lab that's offering genome or exome sequencing, at some level, you have to appreciate that you're engaged in the game of novel gene discovery. And here's just an example from our Rare Genomes project, where we were sequencing a family, 2 SIBs with microcephaly seizures, global developmental delay, and they were non-ambulatory. And in searching through the genome, we found 2 variants in the PPIL1 gene. One was a predicted loss of function variant from the mother. The other was a missense variant, obviously insufficient to implicate that variant in disease, but the data suggests it's very rare to observe 2 novel variants in this gene from our NOMAD database. And so this was suspicious nonetheless. So we entered this gene into the Matchmaker Exchange. And how does that work? The idea is it's a platform where you can put a candidate gene into the system. And then it will search and look for other groups that have put the same candidate gene in. It will bring those matches together, and then it will compare the phenotypes to see if those 2 cases have overlapping phenotypes and might be building valid evidence for a role in disease, or those phenotypes are not a match, and this was probably a spurious observation. Over the years, we've built up this network of databases from around the world. It's actually one of the first federated platforms. What does that mean? It means that there's no central location of all this data. Every database that participates, their data is in their own database, but they allow querying across this network through a common API, or application programming interface. And so anyone can go in, and if you don't have your own node, GeneMatcher maintained by Hopkins, anyone can register an account and enter a gene in there and use the system through that node. And in fact, there's over 15,000 candidate genes entered into Matchmaker Exchange from thousands of individuals, from thousands of contributors from all around the world. So, and we just published a series of papers just last year around the progress of this system in implicating genes in disease. And from just 3 clinical labs that I asked to write papers that I knew were using this platform, they documented over 1,000 novel genes being discovered through this platform. And I can also vouch my research group uses it, and nearly every gene discovery we make benefits from matches on Matchmaker Exchange. So it's just probably one of the most useful tools for implicating new genes in disease that we have today. So if we go back to that case I mentioned, we found those 2 suspicious variants, we entered them into Matchmaker Exchange, and we immediately were matched with an ongoing case series led by Joe Gleason, who had found other cases. Ours was the 8th case. They also had made a mouse model already. And so we actually matched late into that process. Other cases we matched early on and start to build the paper and the publication together. But this is just an example of the power of this system. We were actually immediately able to return a clinical result to that family that we matched on, even though it took another year for the publication to actually come out after going through the review process and all the things that take forever in publications. Now that's sort of where that platform exists today. And in this review that Kim Boycott, Al-Hamash, Danielle Azaridi, and I wrote, we define that as 2-sided matchmaking. 2 groups have a candidate gene, and you're simply bringing them together. But the truth is, there are many cases we all look at when we're doing exome and genome sequencing where we don't find a candidate. Yet we would love to be able to query those data sets around the world. And so we are currently working on what we call one-sided matching, where one group may have a candidate. No one else has identified that as a candidate in another case, but you could query and find that variant in other cases in raw genomic data. And eventually, what we call 0-sided matching, that we can just bring unsolved genomes together in some way to identify commonality through these genomes. But right now we're focused on step B, is this one-sided matching. And we've been bringing groups together through the Global Alliance, where we've now adopted the Beacon version 2 API as the API to allow this new federated network to allow query of raw genomic data sets through a secure portal where none of the data has to be moved into a centralized place like we support a Matchmaker Exchange, but we can query and return limited results. Yes, I have a case with this variant and here's the phenotype, but it's not returning the whole raw genomic data set. So this is a work in progress that we're starting to build to support this latest effort. And with that, I just want to say thank you, and I'm happy to take any questions on the work I presented today or any other questions you want to ask. I'm going to stop sharing my screen and also let my cat out of my office that I discovered once again was hidden in here.
Thank you so much, Heidi. That was such a great talk. We have had a couple of questions.
Great.
But I would just like to start off, whilst I give the opportunity for other attendees to ask more questions. I think your talk demonstrates really amazingly how global collaboration can really improve our understanding of disease gene associations and biology and the way variants cause disease. But I'm just wondering what your experience is on how collaborating on a global scale can help improve our understanding where associations are not so obvious, maybe not so black and white, for example, genetic modifiers, or where variants only cause disease on a certain background, the temporal effects of variants. I'm just wondering what your experience is in that area.
Yes, and I think this is an area where it's sort of that third tier in terms of data sharing, where we're actually sharing whole genomes across different data sets, is going to be critical for us to identify modifiers of disease, because they have a much lower effect in terms of compared to the rare disease, where we often have highly penetrant phenotypes and only a small number of cases are often necessary for implicating Mendelian disease genes. On the other hand, if we're trying to implicate contributions to common disease or modifiers of rare disease where the effect is much smaller, you really need to have much larger data sets. And you're starting to see large data sets get shared, not actually starting. There's been many, like the UK Biobank and now the All of Us Research Program, and many biobanks around the world willing to share their large data sets with phenotypes in ways that can allow us to ask much broader questions and identify variants with much lower effect sizes in terms of their implication disease. So that, so I haven't talked really today about the infrastructure to support those types of analyses, but in a lot of ways it's similar. And the work we're doing within the Global Alliance will allow algorithms to be sent to these large genomic data sets to compute analyses that span many different physically located sets in different clouds to be able to answer these types of questions. So it's a really more sophisticated approach, but something that will enable us to do these other types of analyses.
That's great. Thank you.
Thank you, Heidi. We've got a number of questions now coming in, which is fantastic. I'm just going to pull a few of them off. So obviously, in your today's talk, it's obviously been emphasised for a context, a geographical context. You're talking predominantly about sort of North American developments and its movement from that continent as a hub. But what about a contribution from other continental groups? So Europe, for example, are they quite frequent to the databases, to the work that you're doing? Do you think it would be good to encourage more contribution?
Yes, absolutely. We're starting to see increasing data sets be submitted from other countries around the world. And one of the critical steps, so we wrote a paper early on that said, submitting variant classifications to ClinVar does not require patient consent. And this was critical for our clinical labs to feel comfortable sharing this type of data after doing that work with both NIH and experts in this sort of data sharing experience. Our colleagues in the UK then wrote a very similar paper that also considered the GDPR, which was, of course, critical to understand, the same approaches in Europe appropriate in terms of data sharing, and we're able to define the sort of paradigm as acceptable to submit, variant classifications to ClinVar from European countries, for example. Other efforts have happened in Canada, in Australia, in Africa, and many different places. So it's been a slower to start, and I will say one of the reasons that we got broad adoption in the U.S. is because of a number of not just sort of carrots, but sticks as well. So, one of them is we specifically funded the laboratories and gave them resources because it's not just a question of willingness to submit. It does require work to format your data in a format to submit to ClinVar and then maintain it there. That is work. And early on, my first grant for ClinGen, I had 8 subcontracts to major clinical labs to help, provide the necessary resources to help them get off the ground and figure this out. We no longer fund them, but it was instrumental in sort of getting that started. And I know other countries, that have been supplying and dedicating resources to national clinical labs to help them with this are having better success than those that don't have resources. So there's different approaches, to really try to encourage this critical step, but I'm definitely seeing broader uptake around the world in this case.
Fantastic, thank you. Rachel, over to you.
So I was so engrossed in the answer, I've forgotten to look for the next question. So we have
to answer if you, go ahead.
We've got a question about joining GenCC. So how can a lab become part of GenCC so that they can submit evidence for novel genes?
So anyone can join GenCC. So we have a template to put your data in. So if you just reach out to us, there's an email on the GenCC website, or you can email me, and I'll get you hooked up with Marina DeStefano, who's been the instrumental figure in my group that's been leading a lot of this work. We take in that data and we represent it, and we just ask that you use the standardized framework and some groups have to map their curations to that framework. Others are already using it. But that allows us to harmonize. So there's a critical set of fields that we need, but really we allow anyone to submit into that database.
So we've got a couple of questions here, Heidi, sort of practical questions about participating. So one of them is, does one need to be a clinician to become a variant curator, for example? And do you partner up with industries, and to what extent? And do you have any projects that are broader than just germline, so cancer, for example?
Yes. So you don't need to be a clinician to join. We have many. You have to have a bachelor's degree to be, a college degree to have the qualifications that our FDA application requires to be able to finalize a curation, although we sometimes have interns participating in different ways. But you don't have to be a clinician. So, college level and above are qualified to be able to join our curation efforts. And yes, we have work in cancer, both germline cancer, but also somatic cancer. So we've been partnered with the Civic group. And we now have a corollary sequence variant classification expert panel review committee that reviews applications from our somatic cancer. So we're starting to develop all these different expert panels in different areas of somatic cancer as well, and have a separate committee that oversees that. So we have a great collaboration with our colleagues in that realm.
Thank you.
Thank you. So I've got another question, if that's okay. So I'm just wondering what your experience is in terms of what the biggest barrier to data sharing is. Is it people, IT infrastructure? What are your thoughts about that?
Yeah, it's a great question, and I think it varies, but I do think resources is one challenge, and probably the bigger challenge than simply the willingness to share the data. Now that we find that in the US in terms of querying our different laboratories and why they have or haven't shared. And for example, a few years ago, some said, well, I use a commercial software platform to manage my knowledge, and it's hard to get my data out of that system. So we ended up launching a new sort of resource where we qualify software systems for their ability to support ClinVar submission. And only labs that meet certain criteria have that badge and are listed on the ClinGen website. And we advertise that. It's marketing for the software platforms and encourages them to develop the necessary infrastructure to make this easier for the labs that use those software platforms. So there's different ways that we can help make this easier and reduce those barriers. But I would say that's the bigger barrier in terms of doing this.
Great, thank you.
That was a very interesting perspective there, Heidi, because one of the things I see as a big challenge, particularly in a sort of federated data structure like you're presenting, is the resource. But it's also, these databases cost a huge amount to curate, and you need to be able to have ongoing continuity of curation and database investment, if you like, to ensure that we're going to be able to use them and be able to curate them in years to come. Do you have a perspective on the sort of long-term nature of the resource implications at all?
Yeah, so the challenge that our field has, and if you look at ClinVar, you'll get insight to this. 75% of the variants that have been submitted to ClinVar have only been submitted by one laboratory. Any of us that run clinical labs, if we look at our individual databases, the majority of variants we classify, we never see again. And it just speaks to the rarity of most variation. And so, in some ways, you can't expect a lab that saw one variant once 20 years ago to continually recurate it, right? At the same time, if it's never going to be seen again, it doesn't need to be recurated necessarily until you see it again. So in some ways, more of our efforts on an ongoing basis are focused on those variants that we see over again. And one of the things we found is that in addition to tackling variants that are conflicting between labs where we know we need to resolve it, we also tackle variants that everybody says are uncertain. We all agree, but we don't know what they mean. But by bringing the data together from different laboratories that observed those variants, we're often able to move them up or move them down in terms of classification. So, I think just over time, by sharing all that data, we'll identify those variants that we all have data on, and therefore it's worthwhile to share that data and work to reclassify. We'll also identify those variants that are discrepant, but honestly, the vast majority of variants are just incredibly rare, and we don't necessarily have to invest a lot of resources in those.
Thank you.
And just on the topic of rare variants, I've just seen that we've got a question that's kind of about representation. I'm just wondering kind of what your experience or perspective is on sort of equitable access to healthcare and genetic testing, and how the diverse representation of participants from ethnic minorities is on the outcomes of sort of large genetic studies.
Yeah, it's a great question. So in fact, we have some data. So we just started returning results to participants in the All of Us research program. And there's been a major effort to recruit individuals who have been historically underrepresented in biomedical research. And in fact, 80% the individuals we recruit do qualify as underrepresented in biomedical research. And that's about 50% are based on race and ethnicity, and another additional 30% are based on other socioeconomic factors in terms of geography of where they live, age, different factors, gender, et cetera. And when we started returning results, we looked at the representation of those who were positive in terms of the results we returned on our hereditary disease risk panel versus the rate of underrepresentation in the data set we intook. And what we find is a dramatic drop in the percent that are underrepresented in the positive results going back. That is proof that the knowledge base that we have today is certainly based on an underrepresentation of all communities. And it really focuses our need to address many areas of inequity in genetic testing. Who gets offered genetic tests? Who gets to specialty clinics? These are all areas of our healthcare system that are inequitable today, and it leads to downstream effects in our knowledge bases and our ability to diagnose patients. So there's much work that we all have to do. One of the things that we're doing within ClinGen is, we focus on the variants that are most commonly observed as conflicting or VUSs. But if they're not representative of the full population, we might be not focusing our curation efforts on those variants. So we've been working, for instance, we worked with Invitae to get variants that were specifically identified in minority populations, and then take those variants and cross-compare them to discrepancies and VUSs and ClinVar, and then focus our curation on those variants, even though they're less common, so that we can ensure that our curation efforts are more equitable. But that's just one of many types of work that we all as a community must do to rectify this situation.
And if you were a representative from a laboratory, and maybe a new laboratory in a country that is currently underrepresented, but you had limited financing or resourcing to curate variants in the way that you've described in your presentation, what is the one thing that a laboratory of that type could be doing to sort of help understanding of ethnic minorities and underrepresented populations?
Right. So, I certainly think engagement within the ClinGen community and use of some of the free resources that we use are certainly things that we encourage. So, for example, we've been putting on workshops for our colleagues in Africa, collaborating with H3 Africa and the rare disease program there. We put on workshops to help train individuals in how to curate, train them on how to use our free variant curation interface. We also partnered with our Rare Disease Gregor Consortium to train them to use our Seeker platform, which is a free genomic analysis platform that can be used. So it's a combination of ensuring access to robust platforms that are free to use for the community, teaching them how to share data, teaching them methods and approaches to curate information and bring collaborations together to, which often can result in funded opportunities that bring resources to those communities that can help, really set up for success in areas that are under-resourced.
Great, that's a really thorough answer. Thank you so much. Simon, I think we've got time for maybe one more question, if you have anything.
Yeah, I have. So we've kind of touched upon lots of, the sort of rare disease work, but just taking the model slightly differently. I noticed in some of your slides you were talking a bit more about the pharmacogenetic. There's some evidence from pharmacogenetic variants. Now, historically, we know certainly from our EQA schemes and that there's that the whole curation of variants in pharmacogenetics can be messy. So nomenclature is often very different to what we're used to. So terminology. Could you comment on your efforts to improve clinical interpretation of pharmacogenetic variants? Is that something you're working on?
So we have a partnership with PharmGKB and CPIC. Terry Klein is a PI within ClinGen who's helping lead many of those efforts in pharmacogenomics. And so we have groups focused on that. Some of the terminology for defining haplotypes is distinct. And so we have to work within a system that recognizes star alleles and the complexity there. We actually have some work ongoing within the Global Alliance to help define these star alleles in more robust ways that when they're identified within genomic data can be best represented, as well as defining what alleles and variants within those alleles are actually tested versus not, and assigning the appropriate nomenclature, and also the interpretation of these different alleles in terms of metabolizer state. So there's different terminologies used. So a lot of effort to help, bring consistency and standards to that area of work so that, everybody who runs a genome can apply, different algorithms to be able to identify the star alleles and use the terminology that's understood in the community in those areas for return of results. So it's, there's some way that we need to bridge those gaps. The haplotypes are critical in pharmacogenomics and defining those as allele units, right? So it's a little different, but we are embracing that and supporting that within our community.
Yeah, and it's great. I mean, it's a great answer, but it's also great to see there's a, again, it comes back to sort of more of a federated approach. You are working with those key interest groups to actually support that and ensure we've got some more consistency and standardization as we move forward. That's great to hear. Rachel, I think that's probably, I think we've got time. Have we got time for any more?
I would like to end with a very positive question, if that's okay. So I'm just wondering, Heidi, what you're most excited about for the future of clinical genetics?
Yeah, so I think, that little bit that I shared at the very end, where we're working to enable a federated environment for access to actually raw data. This is a whole new endeavor, but, one of the challenges I find in my work is, just seeing what someone else said about a variant, is challenging. What we really need to do is to share the underlying evidence and data, the cases, their phenotypes, and if we can create a global ecosystem of data sharing, we'll be able to scale genomic analysis and the accuracy of genomic analysis in ways that we just can't do today, where every data sharing bit requires email communication where you say, well, what was the details of the patient you found that variant in? And then they share information they couldn't put in ClinVar, but they have in their own database. And that requires work and effort. It's an enormous bottleneck and barrier to high scale, genomic interpretation, and honestly to application of genomic medicine. So we have to be bold and think about ways that we can share data much more widely and in scalable ways that will allow us to really integrate genomics in every aspect of medicine and research. But to do that, we're going to need to develop new ways to share data that still retain the privacy and security of that data, yet allow more information to be shared. And I just came back from the Global Alliance meeting in London last week, where we're really working to tackle this challenge. And I think that's one of the things I'm most excited about today, is the progress we're starting to make there.
Great, thank you. So I think that's all we've got time for in terms of questions. So I'd just like to thank you, Heidi, for joining us at what is quite an early time in the morning for you where you are. So thank you for giving such a great talk at the start of your day. And I'd just like to thank everybody who's attended today and all the staff at EMQN who've supported the provision of this seminar. And I'd like to let attendees know that we've got another webinar coming up on the 23rd of May, when Dr. Anthony Adamson from the University of Manchester will talk to us about the application of CRISPR-Cas technologies to understand, diagnose and treat human disease. So we hope you'll join us for that, and we'll be circulating the registration link soon. So thanks again. Thanks everybody for joining today.
Thank you, everybody. Thank you, Heidi.
It was a pleasure. Take care.